I'm Patty Reuter-Lorenz, and I'd like to welcome you to the 14th Annual Distinguished Career Contributions Award Ceremony. So this award recognizes outstanding cognitive neuroscientists whose careers are characterized by remarkable contributions across multiple domains of our profession. It's my privilege to introduce Dr. Marie Banich, the recipient of the 2025 Distinguished Career Contributions Award. I don't mind being interrupted by applause, because her career epitomizes the excellence that this award is designed to honor. So let me tell you a little bit about her career. Dr. Banich is the Distinguished Professor of Psychology at the University of Colorado in Boulder, where she also serves as the Executive Director of the Intermountain Neuroimaging Center. Dr. Banich received her undergraduate degree in psychology and biology from Tufts University before heading to the University of Chicago, where she earned her Ph.D. working in the laboratory of Jerry Levy. Upon completing her degree, Dr. Banich was faced with an envious decision: accept a competitive NIH postdoctoral fellowship at MIT, working with Susan Carey, or a tenure-track faculty position at the University of Illinois Urbana-Champaign, one of the very top psychology departments in the country. Wisely, Dr. Banich took the latter offer and soared into an outstanding scientific professorial career, recognized by numerous awards, including a Fulbright Fellowship, a Cattell Sabbatical Fellowship, the Yves and Justine Sargent Prize, and perhaps most fittingly, the honor that we are bestowing on her today. Dr. Banich has an extensive publication record that includes high-impact discoveries about the nature of hemispheric interactions, the organization of executive functions, as well as clinical and applied research addressing how executive functions are engaged in real minds in real-world circumstances. Through this well-funded research program and her generosity as a mentor, Dr. Banich has launched and nurtured the thriving careers of numerous graduate students, postdoctoral fellows, and early career investigators. Dr. Banich will be telling us about some of these contributions today. But in addition to all this, her tremendous research portfolio, in 1997, Dr. Banich published one of the first authoritative and widely used textbooks in the field of neuropsychology and cognitive neuroscience, now in its 5th edition. For 7 years, Dr. Banich served as the section editor on the topics of executive function and cognitive control for the journal Neuropsychologia, and then went on to serve for a 5-year term as the editor-in-chief of the journal Cognitive, Affective, and Behavioral Neuroscience. From 2010 to 2014, she was president of the International Society for Behavioral Neuroscience, and she's been on the governing board of this society since 2019. So these are just some of the highlights of Dr. Banich's many contributions to the profession, and they're set against a backdrop of transformative leadership at the institutions that have been her academic home. As an assistant and then associate professor at Urbana-Champaign, Dr. Banich launched and led the Cognitive Neuroscience and Brain Imaging Group at the Beckman Institute. And this was throughout the 1990s when cognitive neuroscience was a new and emerging field. Then Dr. Banich was recruited to CU Boulder to help launch their brain imaging program, where she also served as the director of the Institute for Cognitive Science, a position she held for over 15 years. In Boulder, Dr. Banich founded and now directs the Intermountain Neuroimaging Consortium, for which she obtained the funding, resources to create and build a neuroimaging facility for CU, University of Denver, and Colorado State. So I could go on and on about the many ways that Dr. Banich has given generously to our field, but let me turn to a personal note about Marie. So our paths first crossed when we were graduate students, Marie in Chicago and me in Toronto, but our friendship crystallized at the very first Summer Institute of Cognitive Neuroscience, Brain Camp, many of you may know, which took place in 1988, I know many of you weren't born then, at Harvard University right down the road. So this was quite an institute, And for historical reasons, it's interesting to share the names of some of the attendees at that institute, including Jonathan Cohen, John Gabrielli, Elizabeth Phelps, Nancy Kanswisher, David Plow, Rich Ivory. We were all students there together. I was fortunate enough to get to know Marie at this 2-week brain camp. We spent hours and hours talking about science, which ultimately established the foundation for a close and enduring friendship. Marie's career and research path has inspired my own, and her insights and friendship have made my scientific journey richer and more fulfilling. And I'm certain that there are many of you out there that feel the same way about Marie. So with that, please join me in congratulating Professor Marie Banich, the recipient of the 2025 Distinguished Career Contributions Award. We have to do this. I'm supposed to take this out of here, and you're supposed to hold it. Okay, there we go. Put this over here.
Well, I have to tell you, I'm just incredibly touched to receive this award. I view the Cognitive Neuroscience Society as my intellectual home, and in that sense, getting an award from my peers, from the people whose research has inspired me and influenced me across my entire career is just an honor that I really, really cherish. So I want to thank everybody for that. And I also really want to thank Patty, who has been my conference buddy for decades, and we've really had some great conversations over the years. So thank you very much. One of the things that I've been impressed about at this conference is the vibrancy of all the younger scholars. And not only do I enjoy the Cognitive Neuroscience Society because meeting old friends, but basically the chance to learn about new and upcoming research. And so, with young scholars in mind, what I'd like to do is just start my talk by telling you that if you don't have it all figured out right now in terms of your career, don't worry. And the reason why I'm starting with that is because I want to just give you some illustration of the way that luck played a large role in my journey and course through my career, some luck that came my way, and a little touch here and there of luck maybe that I took or luck maybe that I made. But I just want to give you a feel. So this is what my career path looked like. Not very linear. And so, as Patty said, I went to college down the road in Tufts University. Why did I go there? I went there because no Ivy League school wanted me. So I ended up at my backup school. As Patty also mentioned, I thought I was coming back to a postdoc at MIT, but I actually ended up getting a job in Champaign, in part because my advisor knew somebody down there who told me to apply, because I was super relaxed at my interview because I thought I'd never get the job. And it turned out there was another very prominent scholar that year who was on the job market, and she didn't happen to apply for the job at Champaign, leaving an opening for me. Why did I end up writing a textbook? Well, I had a colleague who had written a textbook, and he basically had a publisher's rep come and talk to me, and I think in a combination of my naivete and my arrogance, I thought, oh, I'll just take my lecture notes and turn them into a textbook. Little did I know exactly how much work it was going to be, and the publisher made me write the book 3 times before they published it. So I learned how to write. And some other luck came my way when at Champaign, it turned out that they had recently hired Paul Lauterbur, who won the Nobel Prize for MRI. So I sort of got in the ground level with regard to that. Ended up at Colorado. Same story as my first job. They had offered to somebody else the year before. They turned it down. I got it the next year. And some of my work on adolescent development really started with a mystery invitation to come to a presentation at a meeting of the MacArthur Foundation Network on Adolescent Development and Juvenile Justice. So, for all of you young scholars out there, follow your wandering path. Okay, Now, to talk a little bit about my research, I want to give you an idea of the four-part trajectory of my talk. I'm going to talk in the first three portions of my talk about research I've done that has an underlying common theme, which is what we can learn about interacting brain regions and what they tell us for cognition and emotion. And then the last part of my talk, I'm going to be discussing work that I've been doing more recently that's been motivated by personal events that I'll discuss in a little bit. But let's turn the clock back a little bit, and I want to get to the beginning. How did I even become a cognitive neuroscientist in the first place? Well, it's an interesting tale. What I ended up doing was I went to college, as I said, at Tufts, and I went to college assuming that I was going to be a physician, which is why Tufts was an interesting choice, because they had a med school, and I thought, ooh, maybe if I went there as an undergrad, I'd have a better chance of getting into med school. But something happened in my very first class at Tufts. That's me with a bad haircut on the left. You're going to see other pictures of me with bad haircuts. But what happened was I had a free extra slot in my first semester schedule, and I took an intro psych class. And what I learned in intro psych, the professor said that there was this interesting phenomenon whereby left-handers had less lateralized brains. Their two hemispheres weren't as distinct from each other as right-handers. Now, for most people, this probably would not have been a transformative experience, but for me, it was. And the reason was that I grew up on my mom's side of the family with a whole bunch of left-handers, including my mother. Notice what drink we each have our hand we each have our drink in, okay? So I was a poor right-hander amongst a bunch of left-handers who would incessantly tell me how smart and creative left-handers were, and how, my mother's family being Italian, Leonardo da Vinci was a left-hander. Okay? So this kind of piqued my interest, right? There's some relationship between handedness and the brain, and that's the beginning of my career as a cognitive neuroscientist. I went off to graduate school at the University of Chicago, and this was the time of the left brain, right brain craze. A ton of research being done in this area, and the basic idea was, when all the studies were done, the hemispheres process information in parallel, with the left hemisphere looking at the trees and the right hemisphere looking at the forest. The predominant scientific paradigm at that point in time was to pit the two hemispheres against each other and to determine which hemisphere was better, for example, at face processing or word recognition or something of that nature. And the question I became interested in, because of the wonderful work of Roger Sperry, Mike Gazzaniga, and others, and my advisor, Jerry Levy, was what's the corpus callosum doing? Is it this huge fiber tract that merely exists to connect the two halves of our sensory world so we have a unified experience, or is it doing something more? And there were hints in the literature that maybe it was doing something more, because there was some research suggesting that split brain patients had reduced processing capacity, although it wasn't clear whether it was their history of seizures that might have caused that to be the case, or whether there was something else going on. So rather than thinking about the hemispheres in competition, I started wondering about what happens when they dance together. So I decided that I wanted to look at interaction between the hemispheres and test this hypothesis to see if there were some emergent phenomenon from their interaction. Now, the way I investigated this was really relatively simple. Essentially, had a paradigm in which you decide, does the bottom item match either of the top two? And the critical manipulation was whether the two pieces of information required to make the decision went to just one hemisphere, in which no interchange was required, or whether information needed to be was divided across the hemispheres and they had to communicate to reach the decision. So in the first task that I used, it was a simple physical identity task. You just have to decide if the bottom item is perceptually identical to either of the top two. And the results suggested that for this physical identity task, this physical match task, the processing was faster and more accurate when all the information was directed to a single hemisphere rather than divided between them. However, just changed the task slightly, but essentially now made it a name identity task. So I've just changed the bottom letter here, okay? And now you have to decide if the bottom item matches either of the top two. The letters are no longer identical, but they share the same name. And lo and behold, what we observed was that the pattern flipped. Now, across hemisphere processing was quicker and more accurate than within hemisphere processing. And the interpretation we made was that when a task is simple, hemisphere can do it by itself. It takes time to talk to the other hemisphere, and there is no advantage. But as the task gets more difficult, what happens is that division of labor increases processing capacity, leading to the across-hemisphere advantage. So that was my dissertation work at Chicago. But before I leave my time at Chicago, I have to say that there were two things that happened in Chicago in terms of people I met that have had an incredibly large impact on my life. The first person I want to acknowledge is Wendy Heller, there on the left, me once again with a bad moppy haircut. Wendy was my lab mate in graduate school, helped me get through graduate school in many different ways. She has always been interested in the cognitive neuroscience of emotion and is a clinical psychologist and has really helped influence my thinking and my research over the years and has been both personally and professionally a big influence on me. Now, something good also happened to me at Chicago. This is a picture of me with my wife, Laura, on our wedding day a number of years ago. And the whole reason why I met Laura was because I was a grad student doing my dissertation research, and she was an undergrad who needed some spending money. So what she did was she signed up for an experiment I was running, and that's how we met. At that point, I was otherwise involved. We played squash a couple of times, but Laura could be very relentless, I guess had a little flame for me. And she went into book publishing and would come by every 5 years or so and take me out to lunch. And then 15 years later, we got together. We've been together for the last 25 years. So she's been supporting my research since my time as a grad student. And she's been wonderful over the last 25 years, being very understanding whenever I say, "Hi, hon, you know, I rather work than play." So from there, I went to the University of Illinois. And my research initially at Illinois focused on this phenomenon of within versus across hemisphere processing. So the question was this a one-off, or did this really reflect a fundamental aspect of brain organization? And in my work there, I showed that you could find this across hemisphere advantage with higher processing loads across different modalities, across different manipulations of task complexity. And importantly, also, if you put an item on midline and let basically a person either do within or across hemisphere processing, it was optional either way, we saw that at easier task loads, looked like within hemisphere processing was more advantageous, and at more difficult task loads, across hemisphere processing was advantageous. I'd also like to acknowledge some very clever experiments that people in the Cognitive Neuroscience Society did, following up on this work that I actually never would have thought of. Some of that work is done by Patty Reuter-Lorenz, and what she did was showed the implication of this for aging, varying the difficulty of the task. And so if the task is easy, both younger and older individuals show within hemisphere advantage. If the task is hard, they both show an across-hemisphere advantage. But what she demonstrated is that at an intermediate load, elderly individuals switch over to an across-hemisphere advantage earlier, suggesting that with aging, there's reduced processing capacity, and so as an adaptation to that, they engage in across-hemisphere processing. Also, work by Roberto Cabeza subsequently showed that this bilateral field advantage is actually linked to this structural integrity of the corpus callosum, suggesting a strong neurophysiological basis for the phenomenon. So that's where things stood. And then, as I told you, had this serendipitous occasion in which I was in an institution where the implications of this new method of functional neuroimaging became apparent. So at that point, I switched my research career to really look at cognitive control and the roles of the prefrontal regions and their interaction in supporting cognitive control. So there I was trying to figure out, okay, here's this new great functional imaging. What task am I going to use to try to look at cognitive control? And I settled on the Stroop task. I think I've got over 30 publications with various versions of the Stroop task. Why did I pick Stroop? For a number of reasons. I knew it was a robust psychological phenomenon, so that should be a good thing to put in the magnet when you're paying hundreds of dollars per hour. I thought that the Stroop task was also good because it could be used with a variety of populations, young individuals, older individuals, neurologically normal individuals, different clinical groups. Most everybody can name a color. And also because there were lots of variants of the Stroop task that allowed me to investigate control at different levels. What do I mean by that? Well, one, in the Stroop task, there's competition or control required between two processes. For example, what one has to do is override automatic word reading to basically engage in ink color identification. So if I have an incongruent trial, like the word red in blue ink, I have to overcome the bias. I have to exert control to overcome the bias to engage in word reading to do ink color identification instead. But in addition, there was also competition between two representations. If I have the word red in blue ink, I have to decide which one of those two representations, the blue ink or the red word, is task relevant for this particular trial. And finally, there's also competition at the level or control required at the level of responses. So if I have the word red in blue ink, what do I say, either a verbal response or what button do I press, depending on the version of the task. And over a series of studies, what we showed was that different regions of prefrontal cortex played a predominant role in each of these different aspects of control and competition, that posterior regions of the DLPFC and the IFJ were important for the overall task set, that mid-DLPFC was important for control over representations that are now sitting in working memory, and finally, that areas of the medial prefrontal cortex, mid-cingulate regions, were especially important for late-stage selection or selecting which response is appropriate at this time. Okay, so that was my initial setup with my work at Illinois, and then came the millennium, Y2K. For those of you who may remember the turn of the millennium, there was a lot of talk about whether the apocalypse was coming, and everybody was worried that computers were going to crash. But I actually had a nice thing happen to me. It wasn't an apocalyptic change for me at all. But at that point, I moved from the flatlands and the cornfields of Illinois to the flat irons of Boulder, Colorado. So when I got to Colorado, I spent some time basically pursuing a model that there were hints of in the data. And I was interested in what was the relationship between activation in these lateral prefrontal regions and medial prefrontal regions. We came up with the cascade of control model, which basically said that posited an interaction between control being exerted at the level of a task set early on by the dorsolateral prefrontal cortex and that of the anterior cingulate cortex and late stage selection. So the model's really quite simple, but it basically said that if lateral prefrontal cortex does a good job of top-down biasing, imposes a task set, that then there really isn't much late stage or response-related control that needs to be exerted, and so there's not much cleanup, and there's going to be relatively lower activation in the cingulate cortex. On the other hand, if there is poor prefrontal control that provides little top-down biasing, there's a cascade with then late stage and response-related processing that must be done. Control has to be exerted at this later stage before a response can be emitted, and there's more cleanup required. Okay. So I just want to give you a piece of evidence that supports this model. This was work done with Wendy Heller and Greg Miller. And what we essentially did was run people twice through the Stroop task, once in the magnet to provide seeds of these two regions for source localization for ERPs. So essentially what we did was we then were picking up the difference in processing for incongruent and congruent trials over the lateral prefrontal cortex early on, about 300 to 400 milliseconds post-presentation, and wanted to look at the relationship of that activity to later activity in the ACC. So here we're using ERPs because they can give us the time resolution of what's coming first and what's coming second. So what we did was to then test a model of how activation, the ERP signatures, could predict, or the degree to which they could predict actual Stroop interference, somebody's Stroop interference score. So we tested a model that just looked directly at whether DLPFC could predict Stroop interference. That did not turn out to be the case. The model that best fit the data was one of basically the ACC mediating the effects between DLPFC and Stroop interference. And the pattern of results we saw was consistent with the model. When DLPFC showed a high degree of activation, there was very little ACC activity. When DLPFC was low, there was more ACC activity. In addition, armed with this model, we could then look to see whether this could provide insights into different clinical conditions, and this is basically looking at symptoms of depression and anxiety. And what we observed was that people who had higher levels of depression, basically this system through the ACC decoupled, and the direct effect of DLPFC predicted Stroop interference. This result is consistent with, as we know now, the fact that TMS over DLPFC is quite effective for treating depression. Also looked at individuals who have high levels of anxious arousal. We can also think of more commonly referred to as worry. And what we found with these individuals was that it was the ACC activity that predicted Stroop interference, but in an interesting way, that the more ACC activity these people had, the greater the degree of Stroop interference, as if they were spinning around in circles and unable to resolve the late response conflict. We've used this model to investigate a lot of different issues and psychological traits and disorders. I've just listed some of them here. Now, having done this work on the Stroop task, it really got us thinking about this idea of inhibitory control. How do you upregulate one process and downregulate another? And I wanted to move on from Stroop, because Stroop is about essentially perceptual information that we're receiving. I wanted to investigate, or we wanted to investigate in the lab, other aspects of inhibitory control. To do so, we basically turned to a different task, the think, no think task, pioneered by Mike Anderson, which is a wonderful paradigm for looking at inhibition over retrieval of information from long-term memory. And in this task, what we did was essentially did a variation on their verbal version, in which people learn to make an association, a long-term memory association between a cue, in this case in our study of face, and an emotional picture. So in the initial part of the study, people are trained. You make sure that they have learned these pairs. And then in the experimental phase, a cue is shown. And the cue either has, for example, a green box around it, which signals to the participant that they should think about the item that is associated with that face. So think about the car crash. For other cues, there's a red box around it, and that basically instructs people not to think about the item associated with that face, to inhibit retrieval of the associated figure. And importantly, what happens is that people get multiple chances to either try to think of the item associated with those cued faces, or for other faces not to think about the associated item. And then you test them later on to look at their memory. We basically imaged folks during the experimental phase, and what we showed, as Mike Anderson and colleagues had shown previously, is that you get more prefrontal activation for no-think trials than think trials, suggesting that cognitive control is required to suppress retrieval of information. But we were also interested in how is that inhibitory control working? What's going on here? And what we showed was that for the first half of those attempts at impeding retrieval, we saw decreased activation in visual regions of the brain, which support the memory representation. But then in the second half of trials, what we observed was decreased below baseline activity in the hippocampus and the amygdala, which would support the long-term representation of that coupling. Importantly, we found that it was the negative correlation in activation between DLPFC and the hippocampus that predicted the reduced recall on no-think trials. So the more the activity was in the lateral prefrontal cortex and the degree to which it was opposite or associated with reduced activity in the hippocampus and amygdala, the better was performance of individuals in terms of suppressing the retrieval of the item. So at that time, I and my colleagues were fortunate enough to receive a center grant from the National Institute of Mental Health on executive function and dysfunction. And we started thinking about, well, what is inhibition as a psychological construct? And could it be that inhibition is not something unique? It's not a unique process, but it is a byproduct of a general goal maintenance mechanism that allows us to keep the task set in mind. And that inhibition occurs essentially by modulation of target regions from DLPFC, very much in line with some of the ideas that Miller and Cohen had put out. One of the things that made us think about it is that in inhibitory tasks, there's usually very high demand placed on the maintenance of task goals because there's conflicting information, because there's distracting information, because a prepotent response must be overridden. And also leading us in this direction was some work by my colleagues Akira Miyake and Naomi Friedman, who had essentially been trying to look at the psychological structure of executive function. And so they gave people a bunch of different tasks, updating tasks, set shifting tasks, inhibitory tasks. And what they found is all these tasks loaded on a common EF factor, which they suggested was the ability to maintain a goal, especially in the face of distraction. But interestingly, the inhibitory tasks just loaded on that general factor. There wasn't something above that general factor that explained their contributions to executive function tasks. So in the next series of studies, Brendan Depew, a former student of mine, tried to look at this idea of, is there evidence that lateral prefrontal cortex is maintaining a task set, and what it's doing is that it's acting by essentially modulating activity in distant brain regions. The way he did that was essentially to, in the same individuals, look at their performance and their brain activation when they had to inhibit memory retrieval on a think-no-think task, when they had to inhibit an emotional reaction to a picture, you could think of this as a feel, no-feel task, and when they had to inhibit a motor response. And the results basically pointed out that in all cases, it was connectivity from this middle frontal gyrus region that predicted successful performance in the cognitively demanding task, and that there was activation commonly across these three tasks in the middle frontal gyrus. So that led us to conclude that the middle frontal gyrus might play a critical role in all of these three tasks that involves inhibition, and that this region is actively maintaining task goals to then modulate activity in distant brain regions. Now, at this point in my story, it gets a little personal, and I'll share that with you today. This is a picture of my brother Johnny, and he died by his own hand. And as you might imagine, this was an incredibly devastating event for me and my family. And also, personally, because here I was a cognitive neuroscientist, doing work on cognitive control, including in an emotional context, and I was totally powerless to stop the outcome of his death. And in my discussions with him before he died, there was something that was very striking to me. My brother would talk about how he had recurrent thoughts. Now, I want to be clear, these were not thoughts from long-term memory. These were not thoughts of events that had happened in the past, but rather they were thoughts that he said went round and round in his head and that he couldn't purge. things that he was about the hopelessness of the future, or the fact that he wasn't contributing and he was being a burden to people. And so I really began to wonder, how does one control those thoughts? Because I couldn't figure out what to tell him to do to make those thoughts stop. From a more intellectual point of view, I became very interested in this idea because I think it has great relevance for psychopathology. So if you think about most psychiatric disorders, they involve difficulty in controlling the current content of one's thoughts. What varies across the disorders is the nature of those thoughts, but what is common to them is the fact that these thoughts get stuck on replay in the current focus of attention. So I was sitting there trying to figure out, how am I going to study this? What can one do? I looked at the literature, and this is obviously difficult to study, because how do you know when somebody has effectively removed a thought from the focus of their attention? Now, there are a lot of paradigms in cognitive psychology and neuroimaging in which information and working memory is no longer relevant, but most of those paradigms, NBAC and things of that nature, don't really require that you flush or get rid of information and working memory. It's no longer useful, but you don't actually have to purge it. So when looking at the literature, and the only thing I could find was paradigms in which, for example, people were told, press a button when you stop thinking about a particular thought or item. And I found this scientifically rather unsatisfying. I thought maybe we needed to do something else. And I really wanted to know how you can relegate information to the trash bin of your mind. So, for example, right now, I might be thinking about my alma mater, Tufts, and I might be thinking about what weird colors brown and blue are for school colors, and why were our athletic teams named the Tufts Jumbos? You know, I was on the field hockey team. I didn't like identifying as an elephant, okay? So how do we delegate or relegate this information to the trash bin of our metaphorical trash bin of one's mind? Now, in a nod to Ken's talk yesterday, I actually woke up in the middle of the night. I don't know what's happening when I was sleeping that night, but I thought, maybe I can use neuroimaging to address this question. And so in our first study in this area, what we did was to, very simple paradigm, just show people some items, and then item comes off the screen, and you're given a mental operation. I'm going to refer to these as working memory operations. So you could see the Dalmatian, and you're told to maintain it. On the next trial, you could see something like the Clydesdale, and you're told to switch to a peacock. And so there were 4 operations that we ended up looking at and that I will focus on today. Maintain is basically the baseline. And then, in terms of ways to remove information from working memory, we looked at 3 operations: a replace operation, which you replace something with something else, like switching to the peacock from the Clydesdales. Suppress, which you can also think of as a targeted clear, where we tell people to specifically suppress thinking about an item, like that Clydesdale. Or a global clear, in which you're asked to clear your mind of all thought. Suggesting that cognitive control is required when you're trying to suppress or clear information, we once again get prefrontal activation in those regions. Although this initial research was interesting, it still was a little bit unsatisfying. And so in the rest of the work I'm going to discuss today, this has been a collaboration with Jared Lewis Peacock at the University of Texas at Austin, who's been on the journey, our labs have been on a journey together to look at this question. So what we did was essentially decided to use the power of neuroimaging and multivoxel pattern analysis and machine learning to see if we could actually trace the removal of information from working memory. So the way we do this is essentially there's a localizer scan, and we show people a whole series of pictures. And from that, in different categories, we show people an object, we get their brain signature through multivoxel pattern analysis, and we can get then the pattern, for example, that's associated with faces as compared to the brain signature that's associated with places. Now, once we have that, we then can examine how good our classifier is. Are these patterns predictive? And what I show you here is on the left, we have the target category, what they were actually shown. And then this across the top is the decoded category, what our classifier tells us the person saw, or what its guess is of what the person saw. And you can see that these classifiers work quite well. They identify the faces as faces, the fruit as fruit, and the scenes as scenes. Now, this is critical for our approach because this is going to allow us to track how the operation affects the working memory representation. We now know what the pattern of brain activity should look like. So armed with these classifiers, we then apply them to the same pattern I showed you previously, the paradigm in which you show an item, and then you ask somebody to do a mental manipulation on it. What I have here on the x-axis is the category classifier evidence. So higher numbers on the y-axis means that the classifier is saying, yep, that belongs to the category that we think that it actually belongs to. Okay, so what we have here is our first step was to see whether this would work, and indeed, if we look at maintain, and it's the curve shown in green, we found that at the beginning of the trial, it's down at baseline, you tell people to maintain the item, and what you see is a rise in that pattern for that particular category. So just to remind you, we have derived those pattern classifiers for when they were looking at the item, and now we're applying it to when they're thinking or maintaining the item. And we see that pattern instantiated. But the critical issue here was, could we actually see information be removed from working memory? And for that, we had to go to the replace trials. So remember, on the replace trials, you're given an initial item, for example, a face, and you're told to replace it with something else. So if our paradigm works, what we should see is that we should see the information from the initial category, the face, go up, okay, with no evidence for the replaced category, the fruit. But then, as the trial goes on, we should see that the fit, the brain pattern for faces should go down, and the brain pattern for fruit should go up. So we should see a crossover, which is what we observed. So here we can plot and actually watch the removal of information from working memory, which for me was what I had been searching for. And this is essentially my reaction when we got those results. I had a very happy brain. I can't go into detail, but one of the other things we found was that these operations work at different time scales, and also that we can look at, once you remove an item on the prior trial by one of these operations, does it have any carryover influence on the next one? And it looks to us like replace and clear still have, when you've replaced an item or cleared an item, still has an effect on the next trial. We think maybe because it hasn't been totally flushed, but rather you've directed your attention away from it. Whereas suppress seems to be somewhat unique in that it really, when you suppress an item, it seems to have much less influence. It looks like its existence is somewhat erased in terms of influence on the subsequent trial. Now, what we could do is play this same game in terms of machine learning to look at the operations. So essentially, we found that there were different brain signatures for each of these operations: maintain, replace, suppress, and clear. Once again, on the y-axis, we have the actual operation that the person was told to do, and here's the predicted operation. You can see that we can classify. There's a brain pattern that classifies each of these operations, and moreover, and importantly, we showed that you can also come up with classifiers that work across individuals. Matter of fact, we've taken some of the classifiers derived from our individuals in Colorado and used them on a totally unique and new set of individuals at Texas. Now that we have these classifiers, we know that there are different brain patterns for these operations, but where in the brain is this occurring? Can we find information about the neural underpinnings? So in this approach done by Jake DeRosa in our lab, what he did was essentially to take the brain and break it into a bunch of different parcels based on the Glaser parcellation, and then look for each of these parcels, whether characterize the multivoxel pattern within those parcels, 300 or so parcels, and look at the multivoxel patterns for each of the different operations. Okay, so we're looking at how the parcels represent these operations. And then, using a clustering technique, tried to see whether there are certain brain regions that seem to have the same configuration of multivoxel patterns for the configuration of these different operations. And what these results showed was that actually there tend to be 4 different systems, each of which represents these operations in a particular configuration. So, for example, if you look at regions of the brain that are in the occipital lobe or involved in visual processing, what this region of the brain does is it tends to represent suppress and clear, shown here in red and orange, in a similar way. So it kind of treats them as equivalent, and maintain and replace as different, although those two are equivalent. So essentially, these regions of the brain are essentially having an equivalence for those operations in which you actually have something in working memory, maintain and replace, versus those in which you have nothing, suppress and clear. Somatomotor areas seem to differentiate these operations, that they treat the clear operation different from the other 3. If you think about clear, clear is the only operation in which you are not acting on a specific item. Maintain, you're acting on a specific item, replace, you're acting on a specific item, as well as suppress. The regions associated with the default mode treat, maintain, and replace similarly, just like visual regions, but they distinguish suppress and clear. And then finally, the frontal parietal network, which is involved in control, seems to represent each of these 4 operations uniquely. So this gives us an idea, and it's actually rather interesting because it suggests that we have multiplexed representations of these operations. It's not one single brain area that does each of these operations, but the brain actually has multiple ways of characterizing these operations. Now, all of this work, in a lot of ways, has been a lead-up to the question that has really preoccupied me for a long time, and that is, how are these metrics related to difficulties in controlling one's thoughts and in removing information from the focus of attention and working memory? So to examine whether there was a relationship, in our initial work, what we've done is ask people to self-report how much difficulty they have in controlling their thoughts using a variety of standardized questionnaires. We find that these measures are relatively highly correlated, so we use a composite measure across them. And so some people say they have difficulty controlling their thoughts and others don't. And we wanted to look at how these metrics are related, or how self-reported thought control difficulties are related to these brain metrics. And this is what we found. We found that the more someone reports that they have control difficulties, the less precise the differentiation of these operations. Remember, we can look at how clear or differentiated these operations are from one another. They can be more precise, such as the confusion matrix on the left, on the right, or less precise, such as the confusion matrix on the left. And what we found for each operation, so just to orient you, more difficulty in controlling thoughts is higher on the y-axis, less difficulty is lower on the y-axis, and across the x-axis we have precision of the differentiation of these operations, with more precision on the right and less precision on the left. And what we find across all four operations, maintain, replace, suppress, and clear, is that those individuals who have more difficulty controlling their thoughts show a less differentiated pattern, as if they're muddied and they don't have good control over how they would manipulate information in working memory. This gets a little complicated, but we can also look at the consistency of the representations of these operations across the network parcels. Remember, I told you we created these networks, and these are a whole bunch of different parcels that are all representing information the same way. So our hypothesis was maybe not only are these operations not well differentiated, but these networks, the pieces of the networks, aren't all representing the operations in the same way. This is a simplistic analogous representation, but you can think of something of this nature, that it might be that every single parcel is representing its multivoxel pattern. It's all the same across all the parcels for a given operation, or that the representation is variable across the parcels. And so we wanted to see whether less consistent representations of these operations across the parcels are associated with thought difficulties. Once again, more difficulty is up on the y-axis, less difficulty is lower on the y-axis, and across from left to right on the x-axis is less variable representations to more variable representations. We found, for example, within the default mode network that for maintain and replace, the more variable the representations, the worse your ability to control your thoughts. And similarly, for the suppress operation within the frontal parietal control network, more variable the representation, the poorer your thought control. So this also seems to be important, because if we put these different metrics together, we can account for 30% of the variance in self-reported thought control difficulty. So we think that these are neural mechanisms that will be worth pursuing in future research. Now, in some ways, this also was just all a prologue to some of the work that we really want to do, which is to look at how one controls emotional information. So we're in the process of looking at that question right now, and we can compare, for example, scenes that vary in emotionality, a beautiful beach versus a cemetery. And I just want to give you an idea of some of the preliminary results that we've been finding. So these are all initial results with non-clinical samples, but we've found that these operations can be exerted over both positive and negative emotional items, which is actually good news if you want to think about therapeutic interventions. We also have some hints that brain activation may vary specifically in the default mode network depending on emotional valence. So although we don't find huge emotional valence effects overall, there are definitely hints of them. And then the other piece of information that's hot off the presses is we've also used a valence classifier. So a classifier that essentially tells you whether people are looking at a positive item or a negative item. And there's evidence that suppress, compared to other operations, is more effective at removing information about an item's valence, and more so for negative than positive items. Now, I want to remind you that this is about suppression of information in the focus of working memory, not suppression of retrieval of information from long-term memory. Because you may have heard, for example, with regard to PTSD, which is a disorder of retrieval with regards to long-term memory, that suppression is bad. But we have evidence that it may be effective once the content of a thought is in the focus of working memory. Well, where are we going from here? A bunch of ways in which we're following this up. One ongoing study is we want to look at the effectiveness of additional operations. We're looking at, right now, 2 other operations for potentially removing information from working memory, which are inspired, but just the way the shows say, inspired by real events. These 2 manipulations are inspired by meditative and mindfulness practices. And in one of them, we're looking at how effective is the removal operation of focusing on one's breath, focusing internally on the body, versus contrasting that with watching a sinusoidal wave at the same frequency of breathing. So redirecting your attention not to a specific item, but to the external world. In work that Jared Lewis Peacock is doing down at Texas, basically trying to use real-time fMRI to train people to do a better job of implementing these operations. This is why it's important that that classifier generalizes across individuals so people can get feedback on how well they are doing these operations. And as I showed you earlier, when people do not have crisp or clearly differentiated operations, what they do is they tend to have more difficulty, self-reported difficulty in controlling their thoughts. And then, of course, we'd like stronger links to emotional processing and try to explore with people with different emotional profiles, symptoms of depression or anxiety, which of these operations are most effective for a given set of symptoms. Obviously, in terms of treatment, we're not going to be able to put people into the magnet and give everybody biofeedback in the magnet. But if we can figure out which of these operations works well, for example, for ruminative depressive thoughts versus thoughts of worry and anxiety, maybe that can influence and provide insight into new therapies. So that's where we're going in the future. Before I end, I have to say that I have been extraordinarily lucky to be embedded in a network of scientists with whom I have collaborated over the years on many different topics. I've had absolutely wonderful graduate students who have been on this journey with me, colleagues, many clinician researchers who have enabled me to take my work into the realm of more applied issues. And this is, I've tried to, had fun making this diagram, just gives you some idea of the different pockets of research that I've done over the course of my career and the many wonderful and generous colleagues and students that I have to thank for that. I also have to thank my funding sources and different agencies that have given me time to think about all of this. The majority of my funding has come from the National Institute of Mental Health, but I've been very lucky to have funding from a large variety of sources. Now, before I end my talk today, I don't want to ignore the fact that we are in very challenging times. So here I'm telling you, oh, I've had this wonderful career, isn't this great? But these are extraordinary times compared to any I have ever experienced in my career. And I think for young scholars, maybe a little bit daunting. So being a semi-child of the 60s, I'm not quite a child of the 60s, there are some phrases from that time period that I wanted to share with you today to think about. The first is keep the faith, which is to continue to believe in, trust, or support someone or something when it is difficult to do so. I have felt so fortunate to have a scientific career because one of the things I absolutely love about it is it's a way for people to talk to each other. We can have different ideas, and then what we do is run experiments, and we essentially try to find out the common goal. We have conversations with each other. And I think we have to keep the faith that the value of science is just as important as it's always been. And then the other thought is, if you don't want to be part of the problem, then try being part of the solution to encourage proactive engagement in finding solutions. People can get involved in various ways. Some people may gravitate to political advocacy. Other people may gravitate just to telling stories. And I think telling stories about science is important, and talking about what science does and what it brings to our nation. Think about, if you think about what we've learned as a result of cognitive neuroscience, we've learned about plasticity and development and when interventions are important, particular critical times. We've learned about the importance of prefrontal regions, for example, and depression, which has led to TMS, to alleviate depression. We've learned so much about aging and how to keep healthy and development. So sort of spread the word in any way that you can, and that I hope we all hang together and keep the faith. Thank you very much.